Electrophoresis. Electrophoresis is a separation technique that is based on the mobility of ions in electric field. Principle. Different ions have different migration rate depending on their charge, size, and shape. Gel electrophoresis. Gel electrophoresis is a procedure used to separate biological molecules by size. The separation of these molecules is achieved by placing them in a gel with small pores and creating an electric field across the gel. The molecules will move faster or slower based on their size and electric charge. Used for. The purpose of gel electrophoresis is to visualize, identify and distinguish molecules that have been processed by a previous method such as PCR, enzymatic digestion or an experimental condition. Procedure. Steps. 1. Preparing the samples. The DNA is isolated and preprocessed, e.g. PCR, enzymatic digestion, and made up in solution with some basic blue dye to help visualize the movement of the sample through the gel. 2. An agarose TAE gel solution is prepared. TAE buffer provides a source of ions for setting up the electric field during electrophoresis. The weight-to-volume concentration of agarose in TAE buffer is used to prepare the solution. For example, if a 1% agarose gel is required, 1 gram of agarose is added to 100 milliliters of TAE. The agarose percentage used is determined by how big or small the DNA is expected to be. Smaller size DNA bands, higher percentage agarose gel. 3. Casting the gel. The agarose TAE solution is poured into a casting tray that, once the gel solution has cooled down and solidified, creates a gel slab with a row of wells at the top. 4. Setting up the electrophoresis chamber. The solid gel is placed into a chamber filled with TAE buffer. The gel is positioned so that the chamber wells are closest to the negative electrode of the chamber. 5. Loading the gel. The gel chamber wells are loaded with the DNA samples and usually, a DNA ladder is also loaded as reference for sizes. 6. Electrophoresis. The negative and positive leads are connected to the chamber and to a power supply where the voltage is set. Turning on the power supply sets up the electric field and the negatively charged DNA samples will start to migrate through the gel and away from the negative electrode towards the positive. 7. Stopping electrophoresis and visualizing the DNA. Once the blue dye in the DNA samples has migrated through the gel far enough, the power supply is turned off and the gel is removed and placed into an ethidium bromide solution. Ethidium bromide intercalates between DNA and is visible in UV light. Sometimes ethidium bromide is added directly to the agarose gel solution in step 2. The ethidium bromide stained gel is then exposed to UV light and a picture is taken. DNA bands are visualized in from each lane corresponding to a chamber well. The DNA ladder that was loaded is also visualized and the length of the DNA bands can be estimated. Examples of gel electrophoresis. TAE agarose gel electrophoresis is most commonly used for DNA. TBE and denaturing page polyacrylamide gel electrophoresis are common for RNA separation. SDS page is a denaturing gel electrophoresis commonly used for protein identification and separation.